is the compound which is released from moribund cells during the autolysis. We know that plants are continuously, root cells are dying, and this dead tissues or whatever things are coming out, it is generally coming out as a lysate. It contains generally dead organic matter, and dead organic matter is generally not used by rhizospheric organism. It is generally used by saprophytic organisms, or which are popularly known as saprobes. Last one is plant mucilage. Mucilage is one kind of plant polysaccharide, and it is naturally formed from the plant root, and we will discuss its function and its structure and all the things in some later slide. This is Mucigel. Mucigel is a slimy substance that covers the root cape of the root of the plant. Generally it is hydrated polysaccharide, and most likely it is made up of pectin, and which is secreted from the outermost, that is called epidermal cells of the root cape. Now, why it is formed, what is the significance, what is the importance of this mucigel from plant anatomy, that we will discuss right now. Mucigel is formed in Golgi bodies of these epidermal cells of the root cape, and then later on it will be secreted by the process of exocytosis. Friends, you might have heard the word GERL system, and this GERL system, that is called Golgi, endoplasmic reticulum and lysosome system. This system is helpful in exocytosis. Exocytosis means it is the movement of this mucigel or polysaccharides from inside to outside the plant. So mucigel is forming inside the Golgi bodies, and it is secreted out the cell. Now, why it is secreted? Because it is having some functions to perform in root area. First function is coming, that is called protection of root cape, or it prevents desiccation in root area, because we know that generally this mucigel, as we have discussed in last slide, mucigel itself, it is highly hydrated polysaccharide, and if root cells facing any drought condition or any water scarcity or any water severity condition in this area, then what happens, this desiccation condition can be prevented by presence of mucigel. It also serves the function of lubricant for root cape, and it also allow more efficiently to penetrate this root cape inside the soil. Otherwise, sometimes it may cause some mechanical injuries in the root cells, and ultimately it may cause some infections, and then root cap or root cells may also die if such lubricants are not found on the cap of root. Another significance of mucigel, because mucigel is very important exudate, or it is very important substance in rhizosphere, because it provides some sticky or gummy environment for the adherence of microorganism in this rhizosphere. So it creates some symbiotic environment for the fixation of nitrogen fixing bacteria like rhizobium. When rhizobium organisms will come on the root, and when they will make the penetration into the root, at that time, at first sight, they will attach on the this mucigel, and mucigel will help in water absorption. And it also provides a diffusion bridge between the final root system and soil particles, which allow a more efficient uptake of minerals and soil water, or rather soil solution inside the root. So these 5 or 6 important functions are there which are performed by mucigel in this rhizosphere, and they are the main important factor which facilitates the formation of rhizosphere in this area. Friends, these are the densities of microorganisms in rhizosphere. Here some bacterial counts and fungal counts, protozoan counts, and denitrifier and actinomycetes count are shown, and they are counted in form of CFU, that is called colony forming unit per gram of the soil, and they are shown in rhizospheric soil as well as in non-rhizospheric soil. So first column is coming microorganisms, second column is coming rhizospheric count, third column is coming non-rhizospheric count, and the last column is coming, that is R:S ratio, and these figures are particularly shown with reference to wheat plant, that is called Triticum sativum. Now we will discuss microflora of root zone, means what kind of microorganisms are found in this root area. So if you want to study the microflora of root zone, then we are having three specific type of techniques for study of this kind of microflora. They are cultural techniques, then it is coming microscopic techniques, and at last it is coming biochemical techniques. These techniques are useful to study both qualitative as well as quantitative aspect of microflora of root zone, means how many types of microorganisms are there, as well as how many numbers of microorganisms are there. So both are covered under this heading, that is microflora of root zone. Microscopical analysis shows that rhizosphere contains almost all types of microorganisms in it. Bacteria or unicellular microbes are quite common as compared to micellar or filamentous microbes, and in bacteria also almost all kinds of bacteria, they may be size wise or they may be shape wise or arrangement wise, all types of microorganisms or bacteria are found in this area. They may be found either in chain, or it may be also found in individual cell also. Filamentous fungi or actinomycetes are not that much common in this area, because they are having some inhibitory effects in this area. So bacteria are more common. Protozoan are also found, because we know that protozoa are mainly they mainly feed this bacteria. So wherever bacteria are common, protozoa are obviously common, and all types of protozoa like Bodo or Paramecium or Plasmodium, or all these protozoa are found here, like ciliates, flagellates, and all types of protozoa are found in this. Even amoebic protozoa are also there in this area. How to do cultural analysis of this rhizospheric region? Then second method is cultural analysis, and in which plate count is very common method. Plate count is as good as our viable count technique. Here SPA medium can be used, that is called standard plate count agar, or if it is not available, then we may go for our natural or our usual N agar also, that can also be used. R:/S ratio is studied here. R:/S ratio is very important parameter to study overall effect of root on microbial growth. R:/S ratio gives generally good picture of this effect of rhizosphere. Here generally rhizospheric soil and non-rhizospheric soil, that is called bulk soil or fallow soil. Both of these soils are separately taken, and their viable count or this plate count practical is carried out, and then as per the equation n equal to y upon vx, or from colony forming unit, we may come to know which area contains more number of microorganism, and that will give you idea about R:/S ratio will give you idea about rhizosphere is having more prominent effect as compared to bulk soil. Now what type of bacteria are found in this area? Then usually bacteria are having very high R:/S ratio as compared to actinomycetes or fungi or any other mycelial organism. Generally we may encounter gram negative bacteria, rod shaped bacteria, non-sporulating bacteria with respect to root exudates. Most prominent are Pseudomonas, or even Agrobacterium are also found in this area. Not only gram negative but gram positive bacteria are also commonly found, because most of them are saprophytic organisms, they are aerobic organisms. So Rhodes, cocci, spore forming bacilli, Clostridium organisms, they are also found in this rhizosphere. Most common genera of bacteria found in this area, they are Pseudomonas, Arthrobacter, Achromobacter, Alkaligens, Azotobacter, Mycobacterium, Flavobacter, Cellulomonas, and Micrococcus. So we may say that all kinds of positive, negative, cocci, rod, spore forming, non-spore forming, all types of bacteria are almost covered in this area. Not only this type of organism, but even a nitrogen fixing organism as well as phosphate solubilizing bacteria are also more frequently found in this rhizospheric area. Aerobic organisms are usually less found, because as we have discussed in our one of the earlier slide, in rhizospheric area generally oxygen tension or oxygen availability is less, because root is also making the respiration and microbes are also making respiration. So both of these respirations ultimately leads to depletion of oxygen in this region. Bacterial population in rhizosphere. See, qualitative population we have discussed. Now this is quantitative figures of bacteria in rhizosphere. They may ranging from 10 raised to 8 to 10 raised to 9 per gram of rhizosphere soil, and generally 4 to 10% of total root area is covered by bacteria or microorganisms in this area. Microorganisms or bacteria are generally found on root hair and rarely found on root tips, because as we know root tips are having the function to deep penetrate inside the soil, so they may not bear the microorganisms at there, but root hairs are having common these microorganisms. Whatever microorganisms or whatever requirements are there of bacteria, as far as amino acid requirement is concerned, or any growth factors, or even any other growth requirements, these growth requirements and amino acids are they are fulfilled by root exudates or plant exudates in this region, and that is the main reason why the rhizospheric area is this much fertile or this much microbial growth supporting as compared to bulk soil. Now we will cover some of the factors affecting the distribution of microbes in rhizosphere. The first factor is coming, that is called depth of sampling. We know that frequency of bacteria, fungi, algae and other bacteria, usually their number and diversity, both thing is getting decreased as depth of soil is increased, because we know that as depth of soil is increased, oxygen availability is also decreased, as well as availability of organic matter, light and all these things are decreased. So obviously there will be more population is found in surface soil or in subsurface soil, while there is less population is found in deeper region. Second thing is coming, that is plant species. Plant species are also having some influence on rhizospheric flora, because some of the plants are encouraging the growth of microorganism, while some of the plants are discouraging or even they may sometimes inhibiting the growth of microorganism in this area. For example, legumes are having good tendency to support the growth of rhizobium in their area, while grass is having less number of microbes in their roots. Even we may found that biennial plants are having more prominent microbial flora as compared to annual plants, because we know that biennial plants are having the longer lifespan as compared to annual plants. So they are having longer growth phase and there will be more types of microorganisms are found in this area. And third reason is coming, that is called age of plant. Usually young seedlings are also, they are having a positive effect on rhizospheric microbial flora as compared to old age plants. Usually old age plants supports the growth of saprophytic organisms, while young seedlings are having all the metabolic process fast and active, so they are having more prominent secretions and exudations, and that will ultimately leads to growth of microorganisms in this area. This is some physical environment of rhizosphere. Why rhizosphere supports this much growth in that region? The first thing is coming, water potential. Usually plants transpire the water more in daytime as compared to night time or as compared to dark time. So during daytime there will be more water potential is found in the soil, and this high water potential is one of the reason why microorganisms are most commonly found in this area, because microbes cannot survive without water or in dry condition, so they can grow in this water containing area because moisture or water is given by this rhizosphere. Second factor or second characteristic of soil physical environment, that is called soil texture. Usually sand or such type of soil are having larger pores, so they are having more exudate travel space, or we may say void space. There will be more void spaces found in this area, so microbes will get more secretions in this area as compared to some silty or some other large size particle soil texture. pH is also having prominent effect on rhizospheric flora. Here lower pH is found in this area because CO2 level is high, and CO2 is getting dissolved in this soil water, and it will ultimately form carbonic acid and bicarbonates, and that will ultimately leads to lowering of pH, and lowering of pH will also facilitate solubilization of phosphorus and all these things, and ultimately it will give positive effect on microbial growth in rhizosphere. These are the various products which are excreted by normal course of plant roots and which are uptaken by microorganisms, and they will show their growth in this area. All types of amino acid, all 20 or even more than 20 types of amino acids are found in this area. Varieties of organic acids are also quite common in this area, like acetic acid, butyric acid, citric acid, fumaric acid, lactic acid, and almost all tartaric acid, valeric acid, and all. All types of polysaccharides, they may be even pentose sugar, hexose sugar, disaccharide, oligosaccharide, polysaccharide, all types of sugars are also excreted out by plant in this area, and they will be ultimately useful as a nutrient for rhizospheric organism. Not only these carbohydrates and organic acids and amino acid, but even some important growth factors are also very common in this area, like PABA, that is called para-amino benzoic acid, biotin, choline, inositol, nicotinic acid, pantothenate, pyridoxamine, and thiamine. Means almost all kinds of vitamins are also available in this area, so microorganisms growth can be obviously encouraged in this area. All types of enzymes are also found in this area, like amylase, invertase, phosphatase, protease. So these enzymes are readily formed in this area, and they are also available for the growth of rhizospheric organism. And other than all these compounds, some other miscellaneous compounds are also quite common in this area. They may be auxins, glutamine, glycoside, para-hydroxybenzoate, peptides, saponin, and even scopolamine, that is some of the plant hormones, and they also encourage the growth of microorganisms in this area. Now, how plant influence the rhizospheric region, how they facilitate